All right, so the next topic that we are starting with the virus structure, and you will see that some of the material will get a little bit complicated, but we'll have extensive notes in there, so that will help. So the first thing is that why we are studying the virus structure. So you can see that there are just a couple of different types of structures that you're seeing. At the left bottom, you can see that that's an electron microscopic image. That is pseudo-colored. You always get an electron microscope black and white because it's on a phosphor screen. You cannot get a colored image for that. You cannot stain them like a light microscope. And it's pseudo-colored on the top right. On the other hand, what you are seeing, the typical coronavirus on the left, those are the molecular models. And a lot of people think that those are cartoons. The cartoons I have just mentioned in there, the cartoons and the models are very different. Models are atomic structure. We know the position of those atoms, atom by atom. And that's where the structure comes in. And it's amazing that what is the application for that? Say, for an example, in the vaccine development of the SARS-CoV-2, the structural biology played the most important role. And this is what people don't understand is like that, okay, all right, earlier a virus will take, like a vaccine will take 10 years to develop. How can you develop it just within a few months? At that time, the old technology, X-ray crystallography, was used to understand the structures, and you'd go trial and error with this method. And sometimes to solve the structure, it will take 5, 7, 10 years to just solve the structures and do the biochemical experiment. Here, what they did, probably January 10th or 12th of 2020, the whole sequence of the SARS-CoV-2 was uploaded in the GIS. So you know the whole sequence. And then Jason McClellan, he's a biophysical chemistry laboratory in University of Texas in Austin. He was on a vacation. He was skiing in Park City, Utah. And he got a phone call from one of his collaborators from NIH that, hey, this is a new thing. Do you want to work on the spike protein? So that was a funny story. But he just came back to his lab. And from sequence to the structure, it took 65 days. That's it. It's a cryo-electron microscopy, not X-ray crystallography. Purified the virus, and they got the spike protein structure in the prefusion state. That was published in the Science. I have somewhere linked it in there if you need that. So Moderna took that, Pfizer took that, and that's it. You just exactly know that what are the good epitopes when you know the three-dimensional structures. And the Moderna developed the vaccine for the entire spike in the prefusion state, whereas the vaccine developed for the Pfizer is a part of the antigenic sequence. It's not the full sequence. But anyway, so that's the power of the knowing the structure, that how fast you can develop. And this is completely elusive to the mass, that they understand that there must be some hocus pocus that you can just develop a vaccine with that. It is just the technology is very different what it was 10 years ago, 12 years ago. And he was working on the SARS as well as MERS from 2013, 14. So he solved several different structures. And I just probably linked some interviews with him at the Oregon National Laboratory, where they go to the synchrotron to collect their data. Very interesting. All right, so let's start with the basic idea that what are the functions of the structural proteins that make the virus. This is very, very straightforward. The main goal is to protection of the genome. And basically, the assembly of the protective protein shell is important. Just think about that. When you're shipping a material in a box from your Amazon, you need a cardboard box and you need to fold it properly so that your shipment is safe. It's the exact same concept. That's the reason the covering capsid, I just constantly tell it as a box. And the genome and the material that is delivered to you is the nucleic acid that is the present or the item that you have bought, which is delivered to you, is at the very inner part, which is the genome. Sometimes think about that there are wrapping papers or bubble wraps or something like that surrounded around your material. You're going to see that in the structural component, those are some of the what we call as a nucleocapsid protein. There are additional proteins cover the nucleic acid inside the capsid as well. So it's very, very analogous to what you see in a typical package. And also along with that, very important part, this is on the top, specific recognition and packaging of the nucleic acid genome actually take place. So after the assembly of the virus, it is not completely closed. There is a portal open, and there is a sequence that takes place in the cellular environment when the virus is assembling to package the genome and close it completely. It's a really amazing process. And also the most important part is that interaction with the host cell membrane to form the envelope. So you can see right here, in the middle one is a small, say, parvovirus, tiny, small, and a bacteriophage. Those are naked viruses. You do have the protein shells outside. You do not have anything on. On the other hand, the cartoon on the left, that's an influenza virus with the segmented genome. And you can see that there is a roundish envelope, and the spikes that is protruding out from there, those are the H and the N's, hemagglutinin and the neuraminidase. They are inserted into the membrane. So the membrane is that round part right here. And that's an envelope that is derived from the plasma membrane. It could be from the ER nuclear membrane or Golgi as well for different viruses. But in this case, it is derived from the plasma membrane as the virus buds out. And initially what happens, all these proteins are buried inside the membrane, and gradually as the virus matured, they actually span the membrane and sticks out. So that's basically what it is. And the structural proteins have important role because these will then bind to the cognate receptors for the viral entry. Now, of course, if you have a spike like that, it will enter. But on the other hand, for the parvovirus or for the bacteriophage as well, you do not have to have any sticking material coming out of that. Those proteins, that's what you have on the surface, even if it looks flat in the cartoon, they will directly interact with the receptor and get into the cell. So they have that. So the functions, you can see that delivery of the genome is one of the main functions, bind to host cell receptors. And then also they play a role in uncoating the genome. The genome is enwrapped, and the genome needs to go somewhere in the cell. Either it could be the cytoplasm for some viruses, or it needed to be transported in the nucleus for some of the viruses. And then you need to uncoat the genome. And the important part is that the virus need to fuse with the cell membrane. Say, for an example, we're going to see that somewhere else. The virus is taken into the cell, and it forms an endosome. The endosome has membrane around it. So the virus has to, inside the endosome, the virus has to breach that endosomal membrane and deliver the content into the cytoplasm, or then travel to the nucleus. That's where we're going to see the attachment entry in the next part. And then transport the genome in the appropriate site, as we have just said. All right, so here we can see that we are using a word, and very important word, definitely underline that. The virus particles are metastable. So what we mean by a metastable, that it must protect the genome, so it is good. On the other hand, it must come apart on infection or unstable. That's another important thing. So I'm just giving you an example of what it will mean. Say, for an example, your Amazon package is packed in a cardboard box. When it arrives, you can just take a box cutter or something, and you can just cut it open. Now, if your present is shipped to you in a steel case, completely welded, just think about that, how much effort it will take for that to come apart. You have to go somewhere to have a cut that will cut through the steel. That type of packaging is not done the virus. The virus is packed in such a way that it's pretty stable, it's protecting the genome, but when there is a proper signal from the cell, it can come apart easily. That's it. So that's what we mean by the metastable structure. So to do this, this is what it is needed, that it has to attain, for the virus particle to come apart, attain a minimum energy conformation. So most viral particles which are in the environment, for an example, which could be inside an animal in the tissues coming out as well, they have not attained the free energy conformation. So you can see that right here, the first virus is sitting in a stable condition in this valley. Unless it goes to this minimum free energy state right here, it will not come apart. To do this, it has to go over a hump. So this is an unfavorable energy barrier must be surmounted. So how this is achieved? This is achieved usually by a signal that is from the cell. And this signal is usually just binding to the receptor and a conformational change. That small conformational change will make the virus come apart at the plasma membrane or in the endosome or anywhere else. So that's what the important part is. So what happens that when the virus is being made in the cell, in the final assembly part, at that time, just think about that, the virus is just spring-loaded in there. So it is just packed. So you can just think about, again, for the box, that you have a material, you just really put it like it is almost not fitting. Say for the, here the example is, you just put pressure to put that one thing, and then you just sit on it or just put your knee on it and then tape it. It is fine, absolutely. But when it arrives somewhere else that you need to cut it open, if you just use a box cutter, you will see that you do not have to take it out. The top portion will automatically open because the content want to expand. And this is exactly the same concept. So the analogy of the box works very well for this. And the proper signal, your signal for box cutting is a box cutter or anything like that, where here the proper signal is given by the cell from the receptor. That's basically the concept of that whole thing. So here, the metastability is achieved by the symmetrical arrangement of many identical proteins to provide the maximal contact. And what we mean by the maximal contact, that these protein subunits, we're going to define all these terms, are really interacting head to head, tail to tail. So that interaction is very important. But here there is also another, so it identical protein to provide the maximal contact. But here there is a very important thing that you need to know here. The structure is usually, not usually, like almost probably 99.99%, just few examples. They are not covalently bonded together. If you just, the proteins are covalently bonded, you need something to break that bond, like you need a protease or a harsh chemical condition to take those off. So what kind of interactions they are, that's very important. These interactions, it is written somewhere in one of your notes, that these bonding could be hydrogen bonding, this could be electrostatic bonding, this could be van der Waals force, it could be hydrophobic interaction. So if you have a hydrophobic amino acid, say tyrosine or phenylalanine, or these with a large aromatic side chains, they can stack upon each other, the aromatic ring, and that's a very strong bonding among the amino acids in the protein. So just remember that it's not covalent, but all other type which can come apart when there is a proper signal. And here we are looking at a rhinovirus, C15. This is a common cold virus. And you can see that these are molecular models. This is not a cartoon. And how the protein subunits are packed against each other. It's always not the case, but here we are take a look at this, that how this works. So there are three proteins, BP1, 2, and 3. They are in red, green, and blue. And if you just look at the structure, it just is amazing that how densely they are packed into the structure. So you can see that they are head to head, tail to tail. Their surface are kind of like fit in a kind of a puzzle. And due to this fitting, the assembly is spontaneous. It just, when it finds, by the Brownian motion, it finds that groove, it just fits in there. That's it. All these proteins are bouncing around, and they are auto-assembled, because as soon as they find the cognate puzzle piece, they will just interact with them with those forces that I said, except covalent force, and start forming the structure. That's why the assembly is spontaneous. All right. So after that, we are looking at some of the definitions. These are very important definitions. We are going to use that all the time. So the first is that the subunit of a virus. So subunit is a single folded polypeptide chain in its tertiary structure. That means it's properly folded with alpha helices, beta sheets. That's a single polypeptide chain. Now, as simple as a virus like a parvovirus can be built with just one protein, or it could be many protein, but there are some rules we are going to talk about. So the second one is the structural unit. Also, we call this as a protomer or a symmetric unit. So definitely remember that. This is the unit from which the capsids are made. The box is basically made, and this will comprise of one or more subunits. Say for an example, this is again going to the box. If your box is laid flat like at an Amazon warehouse, and if it's folded properly into one box and it is made up of just that piece of cardboard, then your asymmetric unit is just one. That's it. Then if you have two pieces of the boxes and you just put them together to form, then you have two subunits in there. So that's basically the concept of how you build that. So here you are looking at a protein VP1, 2, and 3 for poliovirus. And you can see that these are the three proteins which are interacting against each other in this symmetrical fashion. And we'll go into detail of that. So the protein shell, a capsid, that's the reason I call it a box surrounding the nucleic acid genome. You already know that. And the nucleic acid, so here is a new term, so very carefully understand that. And another term that you use is a nucleocapsid. A nucleocapsid is the core, and here the nucleic acid or the genome, which could be DNA or RNA, as a capsid assembly within the particle. And when we call this as a nucleocapsid, this has to be a distinct structure or a distinct substructure of the virus. What do we mean by this substructure? Say for an example, if you're just looking at this tobacco mosaic virus, it's a non-enveloped naked virus. And the virus is just itself, you have the RNA wrapped with the protein. So it's just by itself, you can think about that this is just a capsid, or the nucleocapsid this is just a nucleocapsid structure. Now, if you have this virus, the similar structure, and if this is your genome for a virus, and it is covered by some protein, the protein and the nucleic acid together is a distinct substructure. This is your nucleocapsid, which is inside the capsid, and sometimes it can be covered by a membrane as well. So it has to be a discrete substructure. And then you have a viral membrane derived from the phospholipid bilayer. As I said, it could be any cellular membrane, but it is always of a cellular origin, and virion is the infectious particle. We already know this. So how the viruses are studied. So electron microscopy, X-ray crystallography, cryo-EM, and the computational power of the cryo-EM is going through the roof. What was not possible by mathematical algorithm to find the structures 10 years back, now it is like unbelievable. NMR we usually don't use. For individual protein components, you can use NMR as well, but not for the virus. They are too big for NMR. So here, there are a couple of things interesting. It's a busy slide. And again, remember that you do not have to memorize any of the poliovirus, pandarovirus, animal cell, these individuals. This is just a conceptual idea that what you are looking at. So here you can see a visual observation under the electron microscope. You can see bacteriophage and a large giant virus. This is what you see. On the other hand, what you are seeing that under this, so electron microscope has the range of about 50 to 70 nanometers. Below that, you cannot really see. So now what happens that after you have that typical structure, you have to do something to find the atomic model, molecular model by something else. And here comes the X-ray crystallography or cryo-EM. So here we can see that at a lower resolution, say for example, right here is a poliovirus 30 nanometer, you will see this. You go up the scale, DNA, this is, you are seeing at 2 nanometer. You are seeing ribosome, this is at 20 nanometer. Ribosome, 20 nanometer. DNA, this is at 2 nanometer resolution. Now, what you will be able to see, the alpha helix in a protein is a 1 nanometer diameter, and you are seeing an alpha helix, part of the alpha helix right here. These alpha helices and the beta sheets are also in the proteins along with that, and the ribosomal RNA, of course. So how you built this model? What do you see actually in a cryo-EM or X-ray crystallography? What you do that we're going to talk about just briefly, that overview of that. You can actually locate each and every atom in the structure, and you basically get this at the top, which we call the mesh-like structure, is the electron density map. So you get the electron density map, and they are very structured. Now, the advantage is that you know all the 20 structure of the amino acid. You just build your amino acid. If you see that in the model, and the computer will do it for you, and you just refine it. If you just see an electron density like this, you know that this is phenylalanine. If you have these two rings, 5-member, 6-member, and 5-membered rings, there is no other amino acid other than tryptophan does have this structure. And see that the phenylalanine and the tyrosine, so the phenylalanine doesn't have something electron density sticking out from here. Here you have an electron density sticking out from the end, and now you know that this is tyrosine, not phenylalanine. So think about that power, that how you can do so. You can build these model into this, and we will come to that how you can achieve these or get these electron densities. So just a couple of overview of that. A nanometer is 10 to the power 9 meter, whereas an angstrom is 10 to the power 9 minus 10 meter, or 0.1 nanometer, 0.0001 micron. So just we will use the angstrom and the nanometer all the time. So just have an overview concept about that. So here is the beginning of era of the modern structural virology in 1940s, and you can see a lot of different structures, really beautiful structures. But think about that you can see the overall virus, but you do not have any structural information. You can just tell, okay, all right, these looks like round. There are some fiber tails at the very bottom. You can see an adenovirus right here. You can see some of the structures, and some things sticks out from there. But again, the molecular detail is not there. So this was not the answer. The resolution was too less. So then we go to the X-ray crystallography. That started, you can see that X-ray crystallography, Rosalind Franklin did all the experiments for the DNA diffraction. Watson Crick almost stole that. She never got recognized and ultimately got cancer. It's a very sad story. But that's how it is. The DNA structure was solved by X-ray crystallography. So here I just put a lot of things, and I'm going to, there will be one day, because we're just going to finish that, and there will be 2 days where we will answer questions, review the material. We can chat about these things in here. So in my first postdoctoral studies, I worked on the vesicular stomatitis virus as a model, and mainly transcription replication, completely molecular biology. Then I moved just next door to Case Western, still in Cleveland, Ohio. And I did some studies with the structural biology. At that time, if you want to do these structural biological studies, you need an X-ray source. Now, the X-ray source, sometimes you have an X-ray diffraction machine here in the physics department. You may have those kind of things. Those are good for some of the material sciences and all those kind of things. But when you are looking at some of these structures, you need an X-ray source which is probably billion or 10 billion times brighter than an in-house X-ray source. And the only place you can go is a synchrotron facility in a national lab. So just that, we used to go to like every 15 days. We used to travel like salespeople. So there were three of them that we used to go. This is in Argonne National Laboratory, 55 miles west of Chicago. And it is called the Advanced Photon Source. That's the picture that you're seeing. Then we used to go to Upton, New York. This is in Long Island. And that was the national, that was another national laboratory, National Synchrotron Light Source, we call it NSLS. And then the other one is is in Berkeley. So it's in San Francisco, up the hill, and they have the they had the synchrotron facility in there. And why we had to travel for that? Because these facilities are used by scientists all over the world. You get a 48 hours or 72 hours time window, 24 hours. There's no 8 to 5 in there to do your experiment. So you go with a group of like 3-4 people. We will have food for those 3 days, and you stay at the beamline for that 72 hours round the clock. You sleep on your seat for hours, and then somebody else will collect the data at that time. Then you just go around. So those were the days. This is like a picture from 17 years ago, probably 17 or 18 years ago in here. This is the beamline that we used to use at the APS. So you can see that it is so a linear accelerator basically inject the electrons into that, and when the electrons go around, there are some beamlines come out of that, and when the electrons lose their energy, they produce extremely powerful X-rays, which is harvested to diffract your crystals. So what we are doing, first, you need to have a crystal. This could be the whole virus. It could be a single protein for the protein structural biologists. If you get the crystal, then when you shoot an X-ray with a small pinhole, it will diffract. So basically, you have the atoms will diffract. And why the diffraction happens, so basically think about that you have an atom, X-ray is going in there, you have a block, there is something in there. That's going to scatter. This is a simple thing, like if you just have a light ray coming in, you can do that. So each atom will scatter the X-ray beam. Now the scattering will happen all throughout the atom, and when they will make a diffraction pattern, basically, and that you calculate, that you electronically collect that on an image, completely automated. So based on that, you build the electron density map, and then you build the atomic model into that process. Now, one of the important things is that we call this a phase problem in the X-ray crystallography. One of these data is actually lost when you collect the data. So what happens is that we collect two things: the position of these dots or spots and the intensity of the spots. That gives you the atom's position in 2 dimensions. There's absolutely no problem. Again, this detail is not necessary. This is just like when you see in other courses as well what it is. But it doesn't tell you that whether this particular X-ray hit the detector at time 1 or time 1 plus 1 nanosecond. So you cannot distinguish that. If you are exposing a frame for, say, even for 1 millisecond or 1 microsecond, there could be X-ray waves that are coming there in a time interval, and that gives you the position of the atom in that direction. You lose that data. You cannot, you cannot do that. So how you do that? Usually what we do that we add some heavy metals we soak the crystals with a heavy metal, and it will bind to a specific site that we know, because you know the sequence of the protein. So it will bind to specific areas in there. And when you now scatter, you know that there will be an anomalous diffraction pattern which is coming from that particular the heavy metal. And using that, you can compute out that in spatial space, where is the phase? You can calculate the phase. Sometimes if you have a similar structure, you can also do something which is called a molecular replacement. So you can just go to this, that the role of National Laboratory in the development of the COVID-19 vaccine. This is not a video, but it's in YouTube, but it's just a talk with Jason McClellan, who was working with the coronaviruses for such a long time, and he was the one who solve the structure of that and how this. So this is what it is. And very interestingly, there are altogether 34 beamlines. They are different things. And there is a BioCat which is reserved just for the industrial purpose. Pfizer and GlaxoSmithKline own that beamline. They maintain that. So there are other academic beamlines as well. So this is how it works. So a lot of extra talk. I just thought this will share because I just get a little excited about that. You can well imagine. All right, so that's it. But again, for reference only, we are not going to ask you these phase problem, how you solve the structures or anything like that. Just have an understanding that how we derive that, so that you are just not blindly looking at structures all the time. So now what happens that the cryo-electron microscopy now replaced that black magic, because it can take 10 years to have a crystal. If you get a crystal, it will not diffract and all these kind of problems. So it's really difficult. Here, you take a virus particle, if it is of the sizable size, and then you just freeze it in vitreous ice, just in water. And then what you do that you just take images by tilting the stage plus minus 70 degrees, and you are getting projection, and you Fourier transform that into a three-dimensional structure. So that's how it is that you are seeing that each projection is a two-dimensional structure, but just when you vary the angle, each time you are getting a slightly different projection. And from that, computationally, you can find by Fourier transforming, you can refine, and by merging those, you can get the structure. So this is a rotavirus structure, and you can see that right here at 20 angstrom, it will look like this. So the background image is a transmission electron microscopy image, and here at 2.9 angstrom, you will be able to see the alpha helices and everything, and you can just go even to the higher resolution, and you will be able to do those. So these are some interesting links that for references only. If you're just interested, you can just directly look at these data. So this is the paper that basically paved the way of development of both the mRNA vaccines, Moderna as well as Pfizer. That's it, period. All right. So after that, the viruses can be classified into three structural groups: helical, icosahedral, complex. So we're going to mainly spend time on the helical and the icosahedral, probably most in the icosahedral, because it is pretty complicated. So let's first take a look at that. And this is the most important thing, that Watson and Crick, we only just think about that their seminal discoveries or not in the DNA structure, but they actually also had a seminal contribution to the virology. And the paper that they here, see that I'm going to talk about, instead of Watson and Crick, I'm going to pick Crick and Watson, because Crick was the first author in that paper. And they were found that the most virus particles are either spherical or rod-shaped. And the idea was that as the virus genomes are small, they are not infinite like in bacterial genome or even close to any other. So particles would be made with many copies of few proteins. And this is what we call as a genetic economy. If you just have a 5,000 kilobase genome, I'm like, there are limited number of genes that you can do fit in there to produce the proteins. So for that, what happens that identical protein subunits are distributed with helical symmetry for rod-shaped virus, whereas icosahedral symmetry for the round viruses. So this concept was game changer, the symmetry concept. So let's see that what it is. So the first is that this symmetry also provides the rules of the self-assembly that we talked about, that how these structures assemble in the cell before it gets out of the cell to infect others. So each subunit has an identical bonding contact with its neighbors, and the repeated interaction of chemically complementary surfaces at the subunit interface naturally leads to a symmetric arrangement. This is like fitting the puzzle that I was talking about. They are similar so that they can self-assemble this way. The rule 2, these bonding contacts are usually non-covalent, reversible, and that gives you the error-free assembly. So say for an example, what happens that if there is something fitting into a space in a wrong way, it's not going to stay in there because it's not very stable. You say, for an example, if you have 2 hydrogen bonds, 3 electrostatic bonds, then something like that, 2 hydrophobic interaction, and there is another structure coming in and just fitting in there with just 2 hydrophobic interaction, it's going to fall apart soon, and it will be replaced by the proper ones. So you need proper fitting, and that's basically what it is, because all these atoms and molecules are moving in the Brownian motion in the cell cytoplasm or nucleus or in the cellular environment, and it will not be stable enough to form a structure if they are not proper. So that's the rule. So here we are looking at a helical symmetry, an example of the tobacco mosaic virus we were talking about. And this just have one protein that enwraps its genome basically. So here we are looking at the subunit, and you can see that the structure is like a corkscrew, and each of these subunits looks like those Dutch clogs. Have you seen those wooden shoes in there? You will find those in some country stores and those. So it just looks like that. So here we have some specific numbers, and you will see a math problem. You will not need a calculator. It will be simple numbers, whole numbers, no fractions, nothing. You will be able to calculate easily. So definitely understand that. So here what we are looking at, that the mu is the 3. This is the number of subunits that you have in a turn. So you have one subunit, so you have 1, 2, and 3. So mu is the 3. The rho is the pitch of a subunit from one end to the other. So that means that if you place these 3 in a plane, here, suppose you have say from the bottom, this is 1 nanometer. So from the bottom, this surface, this top, as it is 1 nanometer, it will be 2 nanometer. This will be 3 nanometer, 4 nanometer. This is the, so basically the subunits are rising. That gives you the cork, like the screw-like, a right-handed screw-like pattern. So you can just calculate the P, and this is the pitch of the helix by just multiplying the rho with the mu, which is 3 nanometers. So you are rising 3 nanometer from here. So that's basically what it is, 1 to 4. So if you are given these values, you can just calculate the pitch of the turn, or you can just calculate. Sometimes we give you this P number, you may have to calculate the rho or the mu as well. So that's what it is. And here is the specific example for the tobacco mosaic virus. You can easily see that. Very self-explanatory. Very simple to understand. Think about this way also, that when you look at a screw that you find in the hardware store, each of them also will have the exact same way of expression. You have a pitch, you have an axial rise, and these are, say, you have a screw like an M4 and 0.7. So those actually denote that what it is, what will be the pitch of the screw and those kind of things. So those are the analogous part if you work with any hardware. So here is a very interesting part, that these are some of the examples, again, of the DNA and RNA viruses with the helical symmetry. And again, you do not have to memorize those, but the take-home message is that the archaeal virus, bacterial virus, plant viruses, there are many examples where the viruses are with helical symmetry, but they're naked. They can be covered by an envelope, but sometimes they may not be. So again, repeating, for the plant, archaea, as well as bacteria, the viruses with the helical symmetry could be naked as well as enveloped. So that's one of the very important things. What we are going to see in the animal, there is an entirely different scenario. Each and every that we know of, each and every virus that we know of with a helical symmetry, all of them are enveloped. There is no naked helical structured virus that infect animals. At least to date, there is none known. So remember that. So that's very, very, very important. So DNA viruses, these are some of the examples of that. And you can see that there is always an envelope, the protein sticking out from there for all of them. So again, you do not have to memorize these. These are just putting that, if you just see them just one time, you will see that the several examples of the cartoons that will just make you familiar with the material. So that's the main goal. So now we are looking at, so right here, that's the thing. And here you are looking at the Sendai virus. And you have the envelope right here. And you can see that the portion of that envelope and the capsid is breached and the nucleocapsid is coming out. So basically what you have, that there are, inside this structure, there is a negative sense RNA. So that's why it is written as minus RNA. And there is an NP subunit. So what is NP? There are two proteins in there. There's a nucleoprotein. There's one nucleoprotein, not two. nucleoprotein, and that enwraps, just like a tobacco mosaic virus, this whole structure. And this structure is packed as a discrete structure inside the whole structure. So this is your nucleocapsid, and you can see this is the envelope around that. So that's what it explains here. All right, so after that, so this is a very simple helical symmetry. So where the RNA is enwrapped with just one protein, and that structure is packaged. Somewhere else, it could be much more complicated. So this is the vesicular stomatitis virus. This is a bullet-shaped virus and of the rhabdoviridae family. This is a model for the rabies virus. Rabies virus, due to the, like it's extremely contagious, you usually cannot work in the laboratory with that. But this will have the same properties in there. An animal virus doesn't infect human beings. That's why you use this virus all the time. So this is the virus I was used to work with. So what we are looking at right here, some of the raw images from the cryo-EM, and this is the solved structure. So what exactly we are seeing in here, if you carefully look at that, let's go from the right towards the left. So here you are looking at that there is a red subunit, and then there is a yellow subunit, and you can see that there is a green structure, which is the RNA, which is inside this. So you can see that this structure, the N protein or the nucleocapsid protein, is the same protein. You have it's just two subunits, just one side. They are all connected, one protein, and they are basically enwrapping the RNA. So you have in just one turn per circle, there are 10 of these N proteins or the nucleocapsid protein that enwraps the RNA. So you can easily see that. And here you are looking at that. So the color, so you have the greenish structure is the RNA. Then you have the yellow and the red. I mean, like this is exactly doesn't translate to that. But that is basically the structure of the nRNA. What is the nRNA? nRNA is basically the nucleocapsid. It is the N protein enwrapped, the RNA enwrapped with the N protein. That is your nRNA. Now, so many of the viruses, say for Sendai virus, if you just look at this structure, this is the nucleocapsid which is put into the envelope. There is nothing in between, that's perfectly fine. So you just put into there. Here, it's a little bit different. You can see that these two shades of this purple and the violet, these are the outer and the inner leaflet of the envelope. Remember that your envelope is a phospholipid bilayer. So you have an outer layer towards the environment and in a typical membrane, and one is towards the cytoplasm. So you have two leaflets. So those two are the outer and the inner leaflets of the membrane. But here is an interesting part, and this will be an exam question, remember that. In between the envelope and the nRNA, which is the nucleocapsid, there is another protein which is called the matrix protein, and that is essential to keep the integrity of the nucleocapsid together. So this is immensely important for the packaging or compacting that nRNA nucleoside. So what happens? Usually in the experimental way, that if we just perturb this structure, what will happen? Say for an example, for the Sendai virus, if I just take the envelope off, you get the nucleocapsid, the picture that we have seen. You get the solid piece of that. Here, if you take the envelope off, the structure is still okay. You can use some mild detergent to dissolve the membrane, and you get the vesicular stomatitis virus, the inside core, surface with the matrix protein. Now, if you use a salt, high salt concentration, the matrix protein actually comes off. It just comes off. Because you know that the interactions are like when you have charged sodium chloride, the electrostatic bondings are broken, hydrogen bondings are broken, come apart. Interestingly, immediately as soon as the N protein comes off, that bullet-shaped structure completely falls apart. That nRNA nucleocapsid will completely extend as such. So to keep the integrity of the bullet-shaped structure of the nRNA inside, the matrix protein is absolutely required. So these are some of the structural implications of additional protein that is required to keep the integrity of the nucleocapsid. That's the take-home message. So here, now we are looking at, so that's all about your helical viruses. Now we are looking at spherical viruses, or and how you basically make that. And this is a little bit more complicated part. So how can you make a round capsid from proteins of the irregular shape? So here is the clue, first clue, that all round capsids have a precise number protein in multiples of 60, like 120, 180, 240, 960, like this. And the second clue is that if you have the spherical virus of many sizes, and most of the capsid proteins are in between 20 to 60 kilodalton, and if they are 20 to 60 kilodalton, then they must be made by an icosahedral symmetry. That means that they need repeated units of these proteins because they are too small. One protein cannot make that whole virus, and they must have this icosahedral symmetry. What do we mean by that? We will explain that in a second. So here you are just looking at just like this is just for the illustration purpose that it shows that poliovirus BP1, this protein, you can see that in a three-dimensional structure is how different it is. We call this a jelly roll. We'll talk about it in a second. But when it is packaged in the virus, the whole virus looks like a spherical. So how this is achieved, that is basically explained by these two clues. So what is an icosahedral symmetry? So an icosahedron is a classic Goldberg polyhedra, which has 20, very carefully with that, which 20 equilateral triangular faces. So you have 20 equilateral triangular faces, and here, let's flatten. So if you take a polyhedron, like an icosahedron, and if you just flatten those, you will get these triangular faces. And let's take a look at animation that if they're folded together, how it becomes the three-dimensional box. So this is your flat box folded into the proper structure. These are the triangular faces, 20 of them, and this is how you fold it into the polyhedra. So that's basically the conceptual idea. All right, so now when you have the icosahedron, and very carefully see this, you will have three axes of symmetry. So what are these three axes of symmetry? So if you look at this right here, you have a threefold symmetry. It's an equilateral triangle, one face, but here you can see that there is a triangle here, there is a triangle here, there is a triangle here. So you have a threefold of rotational symmetry. If you just go straight in this position, you find a triangle, you translate, you find a triangle, translate, you find a triangle. So this is your threefold symmetry right here. If you are here in this one, you get a fivefold symmetry. So you can see 1, 2, 3, 4, 5. These are all equilateral triangle. You have a fivefold symmetry. And if you're here in this position, you have a twofold symmetry. You have a triangle here, you have a triangle here. So everybody good with this? So you have a 3, 5, and a twofold symmetry. If it is just a simple icosahedral symmetry. So here what we are looking at, that you have 20 triangular faces. And what will be the minimum number of protein subunits that is needed to build that structure? That number will be at least you need 60. You need 3 proteins per triangular face. It could be the same protein, and we're going to see that in a structure which are more complicated, it will be multiple. Again, I'm repeating, you have 20 triangular faces, and each triangular face is needed 3 proteins. Altogether, 60, 20 multiplied by 3, 60 proteins in a subunit. So here is a picture. So let's take a look at this right here, where you have a threefold symmetry. You have this structure right here. This is one protein. This is one protein. This is one protein. You have fivefold symmetry. You have 1, 2, 3, 4, 5 proteins. Twofold symmetry, 1 and 2. So what happens here, These interactions of the molecules with their neighbors are identical, head to head, tail to tail. That's how the structure is built. And the particles are basically spherical. They are with the, so the idea is that the symmetry is icosahedral, but in reality, as the curvature of the proteins are different, the structures are spherical. They are not. So you really, there are some of the viruses you can see that, say for an adenovirus, it looks like an icosahedron. But there are many of the viruses, when you look at the structure, it looks like a spherical. But they do have the icosahedral symmetry, but not the shape of the whole virus is icosahedron. So this gives us an interesting thing, and we are going to go into that a triangulation number, and we're going to calculate that triangulation number, which is T. So what happens that if you have a T equal to 1, you have that will make you 60 of these subunits. 60, if you multiply it by the T number, triangulation number, it is 1. 60 multiplied by 1, this is a 60 subunit. And we're going to explain that what it would mean. Now, suppose this structure, simplest one, this we see in the parvovirus, is made with just one protein. And if it is just made with one protein, then you have 60 copies of a single capsid protein. So one of the important things, so you can see that if you just take a one protein right here, here you have a two-fold symmetry, here you have a two-fold symmetry, here this is the axis of a three-fold symmetry, this is the axis of a five-fold symmetry. And this is achieved by, again to have to just remember the name of this topology of the proteins. These are made by a typical topology in structural biology, which is known as a beta barrel jelly roll technology. So, architecture, beta barrel. So, what we mean by the beta barrel jelly roll protein topology. So, you can see that there is strands of beta strands. Those are the flat strands right here. And the next area, there will be flat strands, and they will just bend with a loop and make a flat strand. And that basically makes the walls and the bottom portion. On top of that, it will be again a wall, and on top of that, you will have a wall. And that's how it just encloses the space, basically. Sometimes it's visually difficult. So here we are looking at, say, in the middle of this, you have a five-fold symmetry. You can easily see that right here. So all of these blues are the five-fold symmetry. On the other hand, if you just look at right here in the middle, you have a three-fold symmetry. And then if you just look at right here, you will be able to see two-fold symmetry. So that's basically the difference in between those things. So if you have T equal to 1, which is the triangulation number, you have 60 copies of the single protein. So interestingly, what will happen, here you have these two oranges and one gray. If you have a T equal to 1, you have a single protein. So in a triangular face, these three proteins are interacting together. Usually what you will call that? These three proteins, identical proteins? That's a trimer. That is correct. That is a trimer. So that these trimers are actually very good. The trimers are building this structure. Now what happens is the whole thing that so far I told you gradually start to fall apart. Not completely, but start to fall apart. So I'm just contradicting those structures. If they are larger virus particles, so at that time what happens, these structures are built by adding more subunits. And now you find pentons and hexons. That means that you have, let's take a look at there where you are getting a penton and hexons. You have already 2, 5, and 3, 2, 3, and 5-fold symmetry. Larger structure, anything T more than 1, you will get a 6-fold symmetry as well. So there is a problem. Your 5-fold interaction of the proteins and the 6-fold interaction of the proteins could be similar, but not identical. So that is the thing, that these proteins are interacting in a slightly different manner depending upon their local environment. And this is what we call as a quasi-equivalence. So we are just going to, so again, bonding interactions, all engage tail to tail, head to head, when the triangular faces is made up of smaller identical facets. So what we are looking at here you are looking at this 1, 2, 3, 4, 5 proteins here. You are looking at another 1, 2, 3, 4, 5, 6 in here. The bonding interaction here and here will be different. So that similar, that's why we called as that as a quasi-equivalence. Now, when a capsid contains more than 60 subunits, each occupies a quasi-equivalent position. So only in T equal to 1, when there is a subunit made up of that one, just parvovirus example, that's the only one where you do not have any quasi-equivalence. Anything bigger than that, you have to have a quasi-equivalence. Structural environments are similar, not identical. Now, how do you find the triangulation number? Find the triangulation number, you first have to find a 5-fold symmetry on your structure. So what you are seeing that the H and the K are the directional vectors which originate in the middle or the axis of, on the 5-fold axis of symmetry. So how you do calculate that? So this is the formula of the triangulation number, H square plus HK, K square, and the number of subunits is 60 multiplied by the triangulation number. So that means that if triangulation number 2, you have 120 subunits. If it's 3, 180 subunits, and so on. So let's first look at that, how you calculate the triangulation number. If the triangulation number is 1, you want to go, so remember that from here, you are going just in 1, and right here, this is just only one direction that you are going, from here to here. On the other hand, you can see that this is 2, you are going 1, and then another, you are going like 2 and 1. So you are crossing 2 of these and the 1. So let's take a look at the structure right here. You can see that right here, this is a pentamer and the hexamer. So this is the closest vectorial distance in between the closest 2 pentamers. Remember that, in between the 2 pentamers. So here, if I need to go from this pentamer to this pentamer, remember that all the whites are hexamers. They are not pentamers. The black ones are the pentamers. So what I'm trying to do, that I'm starting from the middle of this pentamer, and I'm crossing how many of these structures? You can see 1, 2, 3, 4 up to this. Then I'm changing the direction into the K direction, in the K direction, and I'm going just 1. So that gives you the triangulation number. So here, center of the start pentagon, move 1 pentagon at a time, and reach the closest pentagon to determine the values of the case of H equal to 4 and K equal to 1. So if you just calculate that, that if it is 4 square, 4 into 1 and 1 square is 21 subunit, and you find the number of subunit by multiplying with the T, so it's 1260 subunits. So here is a YouTube video. You can just easily see that. Basically, the YouTube video is basically based on this, so we will be able to calculate that. So we're just going to calculate very simple equations like this, very, very simple. So here we are looking at some of the differences right here. So we are looking at a structural unit, that's a trapezoid. That's one of the structural unit, means that's one polypeptide, basically. Then when we are looking at a 5-fold axis, this is that structural unit at a 5-fold axis. And here is the capsid, and it just shows where that 5-fold axis is, this whole thing in this position. Here T equal to 1, made up of 60 subunit. Now what happens? The structural unit, in more complex one, the triangular face is made up of multiple facets. So underline that word, that multiple facets is just not only one protein. So you have these multiple facets, and if you just look at this structure, 5-fold symmetry, you have this 1, 2, 3, 4, 5, but you have multiple facets. Here you have T equal to 3 because you have 3 facets here. And so you are looking at a pentamer here. So to build the structure, remember that you need altogether to do the 60, you multiplied it by 5, 5 multiplied by 60, and you will get the 60 altogether. So that's basically what it is. But you can see 3 facets. Here you can see 1, 2, 3, 4. 4 facets, T equal to 4. Here you are looking at in t equal to 13, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So altogether 13. That is per triangular face, you have 13 facets. And this is how a pentamer will look like. And 12 pentamers will basically build the structure. And this is the total number of the subunit, which is the multiplied by the 60t. That's it. So you just multiply 60 by the triangulation number, you will get the subunit. So that's it. All right. So after that, we are looking at some of the structural example of the poliovirus. You can see that it is made up of VP1, VP2, VP3. These are the proteins in the asymmetric, in the triangle. You can see that this is the asymmetric unit. And this is a beta-ballot jelly roll that you can see that it just makes a floor and the wall of that. And on top of that, another structure is put on. So here you can see that the structure in the different areas, they are similar but not, this is a 5-fold symmetry, this is a 6-fold symmetry. So the interactions at the 5-fold symmetry and the 6-fold symmetry along with that is not identical. They are similar but not identical, and we call this as a quasi-equivalence. Now, in SV40, it even doesn't even follow the quasi-equivalence. Here, if it's a t equal to 7, not many examples are there. If it's a t equal to 7, it should be 420. You just multiply by 7 with a 60. But there are 72 pentamers of VP1, altogether 360 subunits. So that means that there will be some space on the surface of the virus that is empty. And that's why it looked like bristle-like structure, because it doesn't cover all of the viruses. The proteins can enclose by some means the genome, but they are not head-to-head, tail-to-tail, and there are some space in there. Whereas you can see that with the quasi-equivalence, the poliovirus structure is much more compact. That's your take-home message. So on the other hand, you can see that large capsid like the adenovirus, it could be get more complex. They are made by these pentons and the hexons, and you can see that this is a hexon trimer and how these proteins are interacting with each other. Even with that, you can see that there are, and with these 12 vertices, those are all good, but even with this, when you have this complex type of structure, sometimes what happens, the penton hexon, this mismatch make an unstable structure. So sometimes what you have to do, you need a cement protein. So you can see that protein 9 is a cement protein which can stabilize these structures. And again, the cement means like it do not glue it, but if it is in there, it makes a favorable electrostatic van der Waals hydrophobic interaction so that the structure doesn't fall apart. So it's you can see that this is the cement protein because the penton hexon mismatch are imperfect and weak structures basically. All of these are happening due to the quasi-equivalence, but it is not perfect. So again, you can see that there is a lot of information in there. Here, this is just for your just for illustration purpose. You do not need to memorize any of these number of proteins or anything. Just if you just remember that protein 9 as a cement protein, I think that should be all right. Here is another very interesting structure, which is the icosahedral protein of the reovirus. It has a double capsid. So you have an outer capsid and you have an inner capsid. So the outer capsid has a VP7 trimer and T equal to 13. So right here, you can easily see that. On the other hand, if you just take out the outer structure, which is the purple one, you get the inner structure with this pinkish color. That has a VP3 as a monomer and T equal to 2. So it's a virus within a virus, and we are going to talk about that, why it is so. Very interesting structure. So this structure goes inside the cell. This structure is made fused with the endosome and delivered in the cytoplasm. This structure never comes off. And the mRNAs are produced inside and just extrudes from these caps. We're going to see that. So rotavirus, on the other hand, is also a Virology family, even have another additional layer. So it is a box within a box within a box. That's basically how you explain that. And we're going to talk about that, why they are so, and what is the functional implication of that somewhere else. Tailed bacteriophage, it is all over the place. So you can see if you just look at the head, it's an elongated icosahedral symmetry. But you can see this, there are many of these proteins you can see, GP23s, GP24s, but these magenta proteins right here, they are not always in a quasi-equivalent position. They are scattered sometimes. So it's very, very complex. On the other hand, although you show of icosahedral symmetry, at one of the 12 vertices, remember that there are 12 vertices on an icosahedron, there is a portal, and that is connected to a helical symmetry, which is the tail. Sometimes it is contractile, helps to inject the DNA material into the bacteria, basically. So here you can see a complex structure with the icosahedron as well as tail structure. Herpes simplex virus, another example. Again, just the take-home message is that there is just one portal. There is just only one portal which helps to package the genome. We call this as a Stargate as well. Another example. These are just a different picture. On the other hand, how much time do you have? We have some time. So now what we are looking at, so these are just examples. See that I just really not going into the detail. You may be thinking that what are the different things that you need to know. So just on the top of the slide, that what you have, that the portal of opening of directly DNA is built on one of the 12 5-fold vertices. There is just only one. Sometimes it is called a Stargate, and this is a portal through which the genome or the DNA is actually packaged inside the particle, and then it is sealed, and it just comes off in that structure. So these are just some of the very, very complex viruses as well. And if you just look at the virus, another pointing out, you have a nucleocapsid in the middle, double-stranded DNA genome, and you can see that there are lots of these proteins. We call these proteins as a matrix protein in VSV. Here, we call them as a tegument protein, and there are more than 20 of them. So in between the envelope and the nucleocapsid, more than 20 proteins are there. Very, very complex structure. So you can imagine that they come in all shape and sizes. These examples are just not for memorizing, just to give you an idea that how complex the structure that they can be. So here we are looking at the capsids now covered with the enveloped virion, and this is a lipid bilayer. We have talked about everything about that. And this is acquired by budding the nucleocapsid through cellular membrane. Could be any cell membrane, that plasma membrane, ER, nuclear membrane, anything. So the nucleocapsid inside the envelope may have helical or icosahedral symmetry. So this is very interesting. So if you just look at that as such, this is an icosahedral symmetry. But if we just go back to what we already studied right here, if you look at the nucleocapsid right here, this is helical symmetry. This is helical symmetry. So nucleocapsid can have all these different structures. All right. So after that, what happens? So we talked about the membrane. Now, the membrane we are seeing right from the beginning is decorated by too many things. So what are these decorations? They are usually always impregnated with these glycoproteins. Almost all of them are glycosylated. They usually have an ectodomain which is sticking out. So this is club-shaped structure which is sticking out. This is cartoon. This is more of this is also cartoon. This is but a little bit more elaborate. Then they do have an internal domain. They actually help in the assembly of the virus, and then they have a membrane spanning domain. And this is your phospholipid bilayer. What you are looking at, that the purple parts are the glycosylation sites, and the green ones are the disulfide bonds, basically. And you are looking at the influenza virus on the left, electron micrograph, and right here. So these are the hemagglutinin and the neuraminidase proteins. So we are just only going to talk about the hemagglutinin because that's the main protein that we're interested in. So here we are looking at the influenza virus. This is a hemagglutinin and then HA. This is basically a trimer, 3 proteins together, but we are looking at just only one for our purpose. So this is molecular model. Remember that molecular model, the membrane spanning domain and the internal domain not shown. It is just only the ectodomain. So the top portion is a globular structure, and this is the rod portion of that. So this is the club club-like structure right here and right here. That's the structure. So you can see that those are sticking out. In some of the flaviviruses, very interestingly, there are some of these proteins on the surface of the envelope. They are really not made up of like this. They don't stick out, but they are lay flat. So here you can see a lot of beta strands, and they are lying flat protein molecules. So we see these in Zika, dengue, encephalitis. Many of the flavivirus family actually show this type of structure. But no matter what, remember that to bind to a receptor, all it needs is a protein. It doesn't need to be sticking out or flat. If it is an interaction going to happen with the receptor, it can happen in any conformation. Then the last part that we are looking at is the complex virus particles. We still have to rely on the electron microscopy at the best and biochemical investigation. We still cannot image at the molecular level. Individual proteins have been crystallized for these proteins, but we do not still have the structure. So sometimes pox virus is made up of, like there's a controversy. Some people think it's just a single membrane or the envelope is covering the virus. Sometimes other groups say that there is a dual membrane covering the virus. So there are some of the complexities in there. Very, very large structures, 350 nanometer in length. You cannot resolve it still with the cryo-EM. On the other hand, for the Mimi virus, you can see that you do have icosahedral symmetry, and in one of the vertices, you have this, we call this as a stargate, through which the genome is actually packaged in there. Very complex structures. So what are the other virion components that what we are seeing? That other than these main structural components, there are many other things that is packaged in the virus. Now remember that if they are packaged in the virus, not necessarily they are contributing directly to the structural integrity which we see, but they must have some kind of minor structural integrity. If they are not packaged into the virus will not properly assemble and eject. So these are also included in the structural part. So what are the things? Enzymes like polymerases. Say for an example, this is a picture of your HIV. So it packs its reverse transcriptase. If it's that reovirus, we saw that before, it contains the RNA-dependent RNA polymerase. The vesicular stomatitis virus is a minor strand RNA virus, contains an RNA-dependent RNA polymerase. So for HIV, it contains integrates associated protein and proteases. Polymerase, which adds the poly tail behind the mRNA. And also the capping enzyme, also topoisomerases. Capping enzyme caps the mRNAs so that when they go out into the cytoplasm, they can be translated. This is, say, for an example, for the reovirus, they will have a capping enzyme function along with their RNA-dependent RNA polymerase. The mRNAs are made within the capsid. When they go out, they are already capped. So they can be recognized by the ribosomes. Then you have activator of mRNA degradation. They can preferentially degrade the host mRNA and shut down the host synthesis, take it over. And also mRNA activator. Along with that, there are some complex viruses which also pack the histones, tRNAs, myristate. This is a lipid. Cyclophilin A. These are some of the host factors also that can be packaged inside the virus. But not direct structural roles, but minor structural role. These are the structural viruses, structural components. Now remember that there was something wrong underneath the notes of this. I updated that before the class, so if you have downloaded it after coming to the class close to 8 p.m., I uploaded it around 7:50, so remember that. So definitely have that. That's pretty much about it. So the non-structural proteins, on the other hand, are the proteins of the virus that is produced in the cell, but never found in the structure of the virus. So that's what it is. So you can see that it is a little overwhelming. So I will cut you here a deal, but sometimes that backfires. That you will not have the quiz due for this on this Sunday. So so you will have time for doing the quiz from the previous chapter. This will be due the next Sunday, along with the attachment of entry. But what happens that people just slack, and then Thursday they're like, oh, we have 2 chapters due, and then do terrible in those 2 quizzes. So the advantage is that you are given the time, so utilize that, because you need to review your material as well. So judiciously use your time. And you will see that the questions will get a little bit more harder and harder. And you will see that some of the questions are recap type of questions, like factual questions. Usually, if I ask a factual question where you need a little bit of memorization in the exams, those usually appear similarly from your quizzes, so that you really have at least seen those questions in the quizzes. You really don't have to do that. But the conceptual question, there will be some that you will need to understand, and those are not memorization type of question. If you understand, you will be able to answer. All right, have a good weekend. And again, if you have any questions, please let me know.